Well, team, we just figured out another way that we can use the microbiome for new and revolutionary things. We're going to talk all about that today on today's episode of Short Stories of Bacteria. So, one of my friends back in graduate school, the way that she always liked to put this was that sometimes bacteria will surprise you with what they're doing. Like, bacteria will show up in some of the most odd places, and maybe we'll rely on bacteria in a bunch of odd ways, but it's never like a really surprising way. It's like, oh, well, I guess that makes sense that bacteria would be involved in that aspect of your life or in that aspect of the environment, or maybe of course, it makes sense that bacteria could be used in this or that way. This is one of those examples, guys. The paper that we're going to be talking about today is one of those examples, and I think it's a really, really cool paper. We're going to talk about that in a second, but first, welcome each and every single one of you guys to another episode of Short Stories of Bacteria. I, as always, am your host, Dr. K, and I'm here to guide you through yet another story chronicling the lives of the tiny and the infamous, and that, of course, is the tiny and the amazing and fascinating bacteria. So today's topic has to focus on, actually, it's related to things like forensics and antibiotic resistance, and of course the microbiome. We're going to jump into that in 2 seconds, but first, don't forget to hit that like, share, subscribe button if you're listening to this on YouTube, or if you're listening to this on some other podcast medium like Spotify or Apple Podcast. Please give us a follow to help support the channel and help support the podcast. But enough of that, let's jump into this paper. So this paper emerges out of, I feel like I should generally have this ready whenever I start talking with you guys, but oh well. Yes, so this is out of the Department of Biology. Shout out to biology at Lund University in Sweden. So good job, Sweden, again with another really cool microbiome paper. I feel like there's always epic papers coming out of Sweden that are related to microbiome studies. But anyway, this one is focused on the microbiome and using the microbiome to track where people are going. So let's contextualize this first. So we've talked ad nauseum on this podcast that every single one of us has a microbiome. I have a microbiome. You, whoever is listening, you have a microbiome. Your pets have a microbiome. We all have microbiomes. All living organisms have some kind of microbiome. And by that, I guess I mean like macroorganisms. And although I guess sometimes bacteria have microbiomes in a weird way as well. But anyway, we all have these different microbiomes. Now, we've also talked about how it's not just humans that have microbiomes, but whole-scale environments have microbiomes as well. So I'm sitting here recording in my office here. My microphone, it has a microbiome. My office has its own microbiome. The handle of your door has a microbiome. Your bathroom has a microbiome. Your shower head has a microbiome. There's a wild paper recently that talked about how there's an absurd amount of diversity on our shower heads just for bacteriophages. But that's a different episode. But the main point being here is that different environments, it's not just live humans that have microbiomes, different environments also have microbiomes. And we interface with those microbiomes, right? So I have a microbiome and I can walk into my office, and then now my microbiome is interfacing with the microbiome of my office. And importantly, these microbiomes influence each other. So every single time that I'm walking into my office, I'm sort of inoculating my office with my own microbiome. And similarly, my office microbiome is inoculating me with the bacteria that are present in here, right? So there's this interface and influence that occurs whenever entities with different microbiomes interact with each other. Okay? Now, in theory, let's take this hypothetical really quickly. So let's say that I have a microbiome, right? And then if someone looked at my microbiome and they found a unique microorganism in that microbiome, right? And this microbiome was specifically located in, on Mount Everest, right? Then you could tell from my microbiome that I had recently come from, or at least been associated with in some way, Mount Everest because of the presence of that unique bacteria. Right? Similar type of thing. Let's say that I run into somebody and I got their microbiome sample, right? And they have bacteria that are specifically present in the Amazon and nowhere else. Well, then I know that in some way this person interfaced with the Amazon, right? The point I'm trying to make here, and maybe you guys can pick up on this already, is that microbiomes, since they're so diverse and so unique, almost act like little fingerprints, right? There's no microbiome that is the exact same across the board, right? And because of this, and because they're so unique, and because we have this interface between microbiomes whenever I interact with my office microbiome, right? Because of this interface and because microbiomes are so unique, in theory, what you could do, right, is you could predict how the species diversity and richness of your own microbiome would change when it's in the presence of a different microbiome. Okay? And if you could do that, then you could use that to figure out where someone most recently was. Okay? So now this is when it starts to click in with forensics. So forensics obviously relies on trace elements of tons of different things. Like they need to have based off of maybe a bit of clay or maybe a bit of fabric or maybe a bit of pollen, you can identify where someone was, say, the night before they got killed or the day before they got killed or something like that. You're able to track where people are based off of these different trace components or trace elements on a person. And so now what these researchers were curious about is like maybe what you could do is instead of using pollen or fabric or whatever, you could use the microbiome of a person to figure out where they had come from, right? Based again, so here's the landscape here. You have a person who goes and interacts with a particular environment. That particular environment has its own specific microbiome. That microbiome influences the person's microbiome, changing it, right? And then when you look at that that person's microbiome later, then you can back calculate to figure out where that person was before, right? It's a really, really cool idea that you could use the microbiome of a person to track where they were before. And anyway, that's what these researchers over in Sweden were doing. And so what they did is they developed something called MGPS, or microbiome GPS. And all this is this basically a GPS system, and using machine learning, they were able to, the game plan was to work towards something that could predict where people had traveled based off of their microbiome. Okay? Utterly revolutionary idea and so cool. Now, they started off with these very, very broad questions like, okay, all right, based off of the microbiome of this individual, can we track back generally to what city they were, where they were located in terms of their city, right? And they actually did a really, really good job predicting this. The MGPS tracked 92% of the samples of these individuals, of these microbiome samples, to the correct city. Right, 92% of these microbiome samples, they were able to pick out, all right, we know exactly which city you're in, okay? And then, and this is where it got really, really crazy, they tried to get even more specific with regards to the location. So, for example, what they were doing in places like Hong Kong or New York, they were trying to figure out which subway station this microbiome had been sampled from. Okay, because it turns out that just as you and I have different microbiomes in my office and my microphone have different microbiomes, different subways in each of these different cities have different microbiomes. So this is all very cool. So what they did, again, they took these samples from these different locations within subway stations in Hong Kong and New York and London as well. And they were trying to figure out, based off of the microbiome GPS, which subway station they were coming from. Now, in Hong Kong, they actually had the best results. They predicted with 82% accuracy, the exact subway station where the microbiome sample had originated. Utterly wild. Now, in New York, they didn't do quite as well. They mentioned that it was due possibly to just the amount of samples that they had. But they were able to predict the right borough of New York for 64% of the time. And this is totally wild, they could even separate out this MGPS. They could separate out the microbiome of just like a kiosk from a handrail going down to the subway. And these two things were just 1 meter away from each other. They were so close, and yet they were still different enough in terms of their microbiomes that the MGPS could pick out whether or not it was attached to the kiosk or the handrail. So cool. So again, let's bring this back out and let's talk about how this would work in the context of forensics. So again, based off of, let's say you have someone who's been killed or something like this, and you don't really have much to go on, but you do have a microbiome sample. And then all you'd have to do is take this microbiome sample and then plug it into something like MGPS, and they could backtrack exactly where that person came from each step of the way, in theory, right? Each step of the way, you could figure out where this person was traveling, and then you could start to pick out, all right, maybe this is where they got killed, or maybe this is how they were transported here, or something like this, right? So cool. But it's also neat because it's not just related to something like forensics, although that's super amazing. It also can relate to things like predicting where diseases come from. So, for example, let's say we have a person who dies because they have some kind of antibiotic-resistant bacteria or something like this, right? And if you have no idea where that bacteria came from, it's harder to combat that disease, right? You're just kind of shooting in the dark as to where it's coming from. And so what you could do then is you could take this individual, and then you could get a microbiome sample, and then you could use MGPS to backtrack again where they came from. And then if you can figure out where they came from, you're able to narrow down the possible locations where this antibiotic-resistant bacteria came from, and in doing so, be able to mount a better offensive against the antibiotic bacteria. So cool. Let's quick wrap this up and then get out of here. First of all, it's important to remember that every single one of us has different microbiomes, right? You and I have different microbiomes, but also our environments have different microbiomes. We have my office microbiome. We have wherever you're listening to this, that has a microbiome. The second point is that our microbiomes influence us, right? So every single time that I go into an environment with a new microbiome, I'm influencing it by inoculating it with my microbiome, and it is inoculating me with its microbiome, okay? Third, because of these differences in microbiomes and because of the influence that microbiomes exert upon each other, we can use changes in the microbiome to predict where someone has traveled. And the researchers from Sweden developed a tool called MGPS, Microbiome GPS, as a way of tracking that. And finally, our fourth, this ability to be able to track changes in the microbiome across time and across space have incredible ramifications in the context of disease prevention, and even forensics and solving crime. So all in all, extremely cool paper. Again, wonderful job, Sweden, for a really cool paper and a really cool tool that can be used now in the context of forensics and science. I hope every single one of you guys had a wonderful time listening to this podcast, and I will see you guys again next week on another episode of Short Stories of Bacteria.